Hello to my dear students. Today in our botany session, we are going to take a very important chapter that will be the biomolecules, right? In this chapter, what you have to take care that in this short session of hardly like few minutes, we have to cover up all the important points. You have to take care of that. While I am teaching you, I will emphasize on many things, right? What are that thing? It will be done if you are listening the full session very carefully, right? So I again repeat my words. This chapter is very important. It comes under the unit cell structure and function. Last time we have discussed cell, the unit of life. Again in short we discussed, but in that short all the main points we discussed. Same like that for the biomolecules. Again we will discuss all the important things, but in a short we have to do that so that all the important things would be covered. Let's start the session. The biomolecules, let's see what are the biomolecules. As the name is indicating, the molecules which are present in the living organism, that you call it as biomolecules. But which type of compounds or the molecules which will be having carbon, or I can say carbon containing compounds, that will be included into the biomolecules, carbon containing compounds. Here we will discuss about the organic substances, not much about the inorganic substances, that we will take only in the beginning for the introduction. So I repeat once again, biomolecules are that substances or the chemicals or the compounds or molecules which are made from carbon containing compounds, right? Like if I say like glucose, it is having the carbon hydrogen together, you can say it is a organic. But if I am saying carbon dioxide, their carbon and hydrogen is not together. Or if I am saying water, H2O, their carbon is not there, only hydrogen is there. So which are not having carbon hydrogen together, that will come under inorganic. Which are having in the formula carbon hydrogen together, you can place them under the organic substances. But this is not a universal truth. Some exceptions are there also for this, but if you keep this point in your mind, I feel many of the problems you can solve from this chapter, right? I again repeat, organic substances which are having in their formula carbon hydrogen together, like glucose, or if I am saying lipids, or in case of the amino acids, these are all having carbon hydrogen together. No doubt other elements are also there, but carbon hydrogen together you consider it under the organic substances, whereas inorganic, where carbon hydrogen are not together or even absent, like N2, nitrogen, this is inorganic because neither carbon nor hydrogen will be there, right? So in this chapter we will talk about carbon containing compounds, means most of the cases we have to pick here about the organic substances which will be coming under the biomolecules, right? Now how we come to know that living organisms are having such type of compounds and elements inside that? With the help of chemistry, means biology or any other subject is not isolated. We need the help of other subjects also, like chemistry here in this subject, where it helps us to analyze which elements are there in a living cells or in a living organism. That is by elemental analysis. Whatever analytic test you are doing, means analysis you are doing in the chemistry, same you have to apply here to access that which type of elements are present. If you will do that in a living organism and compare it with the non-living, you will find that the chemical composition is almost same. In your NCERT book, they have written for percentage weight of earth crust, percentage of these elements, and with the human body, the percentage of the same element. That means what we are made up of, same thing is there in the environment also. But only the thing is that their percentage varies. Now what type of question they can ask from this point? They can ask you the point like which elements are more in percentage in human body as compared to earth crust. When you will see this table now, hydrogen, carbon, oxygen, nitrogen, sulfur, up to here. That will be more in the human body and less in the earth crust. This is the first question. Rest all will be more in the earth crust and less will be the human body, right? So be careful. If you cannot learn the percentage, if you learn, this is well and good, you should learn it. But if not, then have a look on this table the way I am telling you. Next is what? In the earth crust, right? So in the earth crust, when you will see the components, can you see here? The sulfur and the carbon, they are in the same percentage. This is the next question. Which two elements will be in the same percentage in the earth crust? That will be carbon and sulfur. Next question, what they can frame? Like, can you see the nitrogen is very little in earth crust, but silicon will be negligible in the human body. They can ask you this question, what is negligible in human body? Silicon. What is very much less or little in earth crust? That will be the nitrogen. Likewise, that type of question will be framed from this table. And do remember, this is table like 9.1 in your NCERT book. And read it like table 9.1, not figure 9.1. Figure 9.1 is something else, which is containing these structures. Are you getting that? So this page is telling about the all organic molecules, what essentially an organism will have. Few examples they have listed here. So be careful, this is figure 9.1. But when you will see in your NCERT book, this is the table 9.1. So read your lines carefully in your NCERT. So this is a set of questions what they can frame from this table. Now this is all done by elemental analysis. Now if we want to check on the chemical composition, that which organic and inorganic substances are present in living organism. I said you biomolecules are the carbon containing compounds, but it never means that inorganic is not there. Inorganic is even there. That together you call it as cellular pool. Let me to say this point for you again. Carbon containing compounds, I said you that you call it by the name biomolecules, right? But if I will take these all biomolecules plus other compounds or other components which can be like ionic form, right? Like minerals are there, right? Even water, gases, that all together you call it as a cellular pool. This all together you call it as cellular pool. So cellular pool is bigger thing under which biomolecules will come, where we will talk about carbon containing compounds, means most of the organic substances we will talk about. But it never means they don't have inorganic. They will have inorganic also, which will include mainly these substances. These are inorganic substances present in the cell of an organism. Is that clear? Now elemental analysis will give you an idea of elemental composition, and we can compare it with the element composition by percentage of weight with earth crust also, if I take the example of human body that I discussed, what type of questions they can frame. Now about the chemical composition, how we can understand the chemical composition, how to analyze the chemical composition of the living organism, that what type they are having. For this we are having chemical analysis. For chemical analysis, what we have to do? We have to take a sample from the plants, like some vegetables in a form of these vegetable, or like even the leaves also, like spinach leaf we can take, or if animal you are taking, then best source is the liver. And what we have to do? We have to take the acid here. Which acid? Trichloroacetic acid. This is next important question. Trichloroacetic acid we have to take. Cl3CCOOH. This is a molecular formula of trichloroacetic acid. This is very important, that during chemical analysis, which acid we are using here, trichloroacetic acid. So first of all, we are doing the grinding. Grinding is done in which medium? Acidic medium, trichloroacetic acid we are putting and we are grinding it, right? And by which thing we are grinding? This handle like structure that you call it as pestle, and in which we are doing like a ball like mortar. This is a common apparatus in our kitchens also. So it is very simple to do the grinding, but the medium here we are using the trichloroacetic acid. After doing all these grinding, we are getting a thick slurry, right? We are getting the thick slurry. Don't look to the picture right now, just focus on what I am saying. After grinding, we are getting the thick slurry, and that slurry we have to put on the filtration process. Now we have to focus on the filtration. After grinding in pestle and mortar, now we have to filter. Can you see on that top we have kept the cloth, which you call it as a cheesecloth, or even we can take the cotton also to filter that. When you will filter, it is same like when you are filtering a tea. The tea leaves which are not soluble, it will remain on the sieve upper side, and which is going to be soluble in water and milk together with the sugar and tannins from the tea, all will dissolve. That will come in a cup in a form of filtrate. So can I say after filtration you will get two fractions. What are that two fractions? Upper side we will get the retentate, and on the lower side we will get the filtrate. This is the next important question. Please do listen it very carefully. Elemental analysis based on different elements, what they are present. We are taking help of chemistry to analyze that elements. But now which compound, organic, inorganic, which compounds are there? For this we are doing the chemical analysis. There we use the process of grinding, then filtration. After filtration we got retentate and filtrate as a fractions. Now let's see after analysis what we will find in the retentate and filtrate. Obviously, which are insoluble, which are going to be insoluble in acid, that will come in the retentate. But which are soluble in acid, which is soluble in acid, it will come in the filtrate part. Now after this process there will be the analysis test, there will be the separation technique. What we need we will separate out. But before that, let's see which insoluble substances will come in retentate, because you have grind the living cells. So in that living cells there are membranes also, you disturb their membranes also, which is made up of lipids and proteins. So in the cells even other type of macro micro things are even there, which you have grind and put it into the form of the slurry. And after the filtration, slurry will divide into two fractions, retentate and the filtrate part. Again I repeat, retentate is insoluble in acid, filtrate is soluble in acid. And when you will check, retentate will include only the organic substances. Only organic substances will be there. But in filtrate you will find both organic and inorganic substances. You will find both, right? Now let's see which organic substances in retentate you will get. It will contain the macromolecules, mainly are the macromolecules, and only one micromolecule will be there, which will be the lipids. That will be the micromolecules. Right? Now let's talk on this point, what is macro and what is micromolecules. As the name is indicating, macro definitely will be of bigger size, micro will be of the smaller size. So, and if it is bigger in size, obviously their molecular weight will be having the higher value. So what value they are having? It will be having molecular weight more than 10,000 Daltons. But micromolecules will have between 18 to 800 Daltons. Means less will be there for micromolecules. Lipid is having even less than 800 Daltons. That's why you call it as micromolecules. But where it is coming? In the retentate, in the insoluble fraction of the acid, or you can say insoluble fraction of the filtration process, it is coming as a retentate. Fine. So do remember this fact. This is important. Retentate will only contain organic substances. It will be most of the macromolecules, and only one micro will come, that will be lipids. Lipids are having weight less than how many Daltons? 800 Daltons. But if you will see the filtrate, it will contain both organic as well as inorganic substances. Right? I have already mentioned that thing. So better to save the place here to write another point. I repeat from here. Filtrate include both organic and inorganic. Now, is that a macro or micro? It will be all micro. All will be the micro form. Micromolecules. Except what thing? Lipids. Except lipids. Because lipids you have collected in the retentate form. Right? Now, let's check on that here organic matter will be which one and which one will be inorganic. Let me use the next slide. In the filtrate, I am taking for filtrate. I said you it is having both inorganic as well as the organic substances. Inorganic, I already mentioned you, water will be there. Even it is having the ionic forms of the minerals or other neutral forms will be there, and add on to gases, but gases are going to be released, right? So mineral include ionic forms also, right? Organic will include what thing here? It will include like amino acids, right? It will include the lipid, not lipid here right now. Lipid already comes into the retentate. So here I must write for sugars, right? And even it will include nucleotides. Even it will be including for the like vitamins, and even for some hormones which is going to be there, right? So main will be the these part. We will take these things main, and rest of the thing depend upon their molecular mass. If it is more, it will come in the retentate. If it is less, then it come in the filtrate form. Obviously which are smaller, it is more soluble, but only one, that will be the lipid, that will come along with the macro form. Reason why? Like suppose you are having the oil, you are putting a drop of oil in water, will it mix there? Not. Same will happen with the lipids in the trichloroacetic acid. It will not dissolve. It will come like a vesicles, like a droplets on a water of oil. Like that it will collected in a form of vesicles in the retentate fraction of it. Getting that? So this is very important thing. In filtrate you will not get lipids. Although lipids are micro, but rest all micro form which are less than 800, or you can say I must say less than 1000 Daltons. They are less than 1000 Daltons. It will come under micro. So almost all micro are here, except what thing? Lipids. Lipids will be collected in the retentate, and why it is collected? Because it is insoluble into the trichloroacetic acid. They will come in a form of vesicle. Like this we are analyzing what things are there in the cell. Then chemistry will play its role, that you have to put the separation technique, then you have to purify that, only then we will collect the respective components out of it, right? Now the topic is about biomolecules, so we will restrict only on the organic molecules. Either it is a macro form or it is a micro forms. So we will constrict our lecture only on the biomolecules, organic one, fine. Now what we have to take, when will we go with the composition? It is not like that. We are not analyzing the complete story. The complete story analysis is in this table. What it is telling you about, that either organic or inorganic, if we compare from the extract based on the percentage of the total cellular mass, who is having highest percent? Water, 70 to 90%. Proteins 10 to 15%. Then carbohydrates written 3%. But don't say that after protein less will be carbohydrates. After protein it is nucleic acid, 5 to 7%. Do take care of this thing. If they ask you third in the list will be the nucleic acid, not carbohydrates. After nucleic acid, carbohydrate, then lipids, then ions. Which type of question they can frame? Arrange this percentage in increasing, decreasing order. Next thing, if I ask you, which among the following is organic? Proteins. So if they ask you who is having, means which organic substance is in highest percentage in the cellular mass, then your answer will be proteins. Then don't say water, because water is inorganic. Is that clear? So just take care of all these things, and that only one analysis I discussed with you, that is chemical analysis. If we do the ash analysis, this is more for inorganic substances, like this table given in your book, where they are talking about the ionic forms and neutral inorganic forms. What is that? Sodium, potassium. These are monovalent cations. Calcium, magnesium, divalent cations. Water is universal solvent. And compound which are neutral, sodium chloride, calcium carbonate. Complex type of ions, phosphate and sulfate. At this point, take care of their valency. Phosphate with the 3 minus, sulfate with the 2 minus. So such type of questions even they can ask, which one are the monovalent cations, sodium and potassium. Divalent cations here in the living tissue composition, calcium and the magnesium. Water is a neutral, which is a universal solvent. Sodium chloride, calcium carbonate, again neutral, minerals over there. And complex type of ions are phosphate and the sulfate. So I am not saying only this, but as per NCERT, you have to go with these information, and this way they can ask you the question. Now coming to the next part, just now I discussed the analysis, but do remember one more table is very important in this chapter, that is about the secondary metabolites, right? Now to understand what are secondary metabolites, we have to compare it with the primary metabolites, right? We have to compare the primary metabolites from the secondary metabolites. See the word meta means what thing? Change. Whatever biomolecules in our body, it is not permanent. It has to change from its one to the another type. Like for example, you are eating the glucose, or glucose is entering into the cell. Let me to start from there. When glucose enter into the cell, it will not remain glucose. It will convert into glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-bisphosphate, so on. Glycolysis will happen. So many steps are happening over there. If you are intermediate student, just started with +1, don't worry, just think about what we are eating is not permanent in us, that keep on changing into one form to another. So these biomolecules, you can say they are the metabolites which keep on changing from one form to another form. So these metabolites, either micro, macro, we are not talking about, that is another category based on their mass, we already discussed. But now this is another classification of that metabolites based on how much it is important to the cell or by the host it is secreted. For example, if I am telling you turmeric we are using to cure with some of the like some injury happen, sometime we are applying turmeric over there. So can I say turmeric is useful to us? Because it is having curcumin, one of the drug inside that which can cure us and help us as an antiseptic. But curcumin, how it is helping to the turmeric plant, we don't know. That type of metabolite you call it as secondary metabolite, right? So that metabolite where their role direct as a primary metabolite, you don't know, that type of metabolites will categorize under the secondary metabolites, right? Primary are what? There you know the role, what they are performing, like nucleotides, are becoming the monomer of nucleic acids. Or if I am saying glucose, it is again the monomer of the cellulose, or even for the glycogen, even for starch. They are the primary metabolites. They will break and give us the energy. So for the primary metabolites, their primary role is known. But for secondary metabolite, their main physiological role is not known to we people. But definitely they are useful for human welfare. They are for human welfare, and some are even there which is having ecological importance. Such type of metabolite you call it as secondary metabolites, and that will be given by, means what is a host who is giving us that thing, that is by the plant. Some plants will be there, fungi will be there, and other microbes will be there, right? So I mean to say here the word host. Host means the source from where we are collecting. Like for example, penicillin is good for us, but how it is penicillin is useful to the fungus, penicillium, we don't know. It will come under the secondary metabolites, right? So likewise, in your book, they had given one table of these, the main groups of the secondary metabolites and their example. Please do learn that. This is very important. We are finding this question many times in the pre-PYQs. So go with that also, right? Now, if I want to give you the overall view of the molecules present in the living organism, how I can make it as a note for the remaining few minutes what we are having, and we should cover all the important aspect. Let me to say, we discussed chemical analysis. Let's a little bit recapitulate the point. In chemical analysis, we got retentate and filtrate. In retentate, majorly we are getting macro forms. Only one micro is there, which are lipid. So we have to discuss all about all the points of the retentate. Then for the filtrate, we are getting inorganic, organic both, but micro forms, micromolecules of that, right? So I am not taking inorganic because we are talking only about the biomolecules. So our remaining topic, what we are left with, there we will discuss about all that organic stuff, what we said is present in retentate and filtrate, one by one. And do take care of the two words in the discussion. One is that polymer, and second is what? It's monomer. Are you getting that? Polymer is the long chain of the small unit. That small unit we call it as monomers. Like suppose we are having garland of flowers. So can I say full garland is a polymer, and each flower there, that you call it as monomer of it. So what we are having in micromolecules in the filtrate, many examples are there which is going to be the monomer for the macro form which is in retentate. How? That we are going to discuss, right? So in that way we can discuss the chapter also. Like if I am discussing about the micromolecules. So micromolecules in this chapter, they had discussed about like amino acids, even they had discussed about the lipids, and even about the nucleotides. Somewhere little bit we have to say about the sugars also under the carbohydrates, right? So let's see one by one all. If you know about their micromolecule form, then we can talk their polymeric form also. But do remember, lipids will always be remaining in the micro form. They don't have in their macro forms of it, right? So I will discuss that which monomer is forming which polymer. So here right now, amino acid, nucleotides, sugars, these are becoming the monomeric form of their polymeric form. Sugars are actually be divided into the two main categories. One is the monosaccharides, one will be the monosaccharides, and next will be the oligosaccharides. Right? They are sweet to taste, but their polymeric form are not sweet to taste. That's why you cannot call them as sugars. Means under the carbohydrates, two forms are there, micro and macro. Micro forms you can call it as sugar, being it is sweet to taste, but not the macro form. Like macro form example is cellulose. If we eat the cellulosic sources, it is not sweet to taste, but if you eat the glucose, which is a monosaccharide form, obviously it will be sweet to taste, right? So, but these are the micro forms. But as a monomer, what is there? Monosaccharides will be the monomers for the polysaccharide form, right? Now let's see amino acids first of all. So let's talk about their polymeric form, then I will discuss one by one all the points. Like amino acid, their polymeric form you call it as a proteins, right? And how they are forming? This is formed by peptide bonds. They are formed by peptide bonds. Lipids, I said you, it will remain as a micromolecule form. So what is there on this side? That will be the macromolecules. Do remember that. Although they are polymeric, they will come under macromolecules. Lipids are not having any their macromolecule form. Nucleotides will forming which one? Nucleic acid. Means what is a monomer of nucleic acid? Nucleotides. Now nucleic acid, how it is formed, by which bond? That is by phosphoester bond. Right? Then after that sugars, if I am saying monosaccharide, what macro form they will form? Polysaccharides. Right? Polysaccharides are formed by which bond? By glycosidic bond. It is formed by glycosidic bond. Fine. Let's say one by one. Let's check on amino acid, how this happens. Now see, if I am taking amino acids, they are the substitutes of the methane. Methane structural formula you are writing like this. This is for methane. If I replace one of the H group with the COH group, carboxyl group, one with the another with the amino group, one H remain as such, other will be by variable group. Can I say one same carbon is surrounded by different groups? That's why this carbon you call it as alpha carbon, which is the first carbon if you are taking R as a long chain. Alpha and surrounded by different groups. Being here we have carbon alpha, for all amino acids you call it as alpha amino acids. There are hundreds of the amino acid, but hardly 20 amino acids are there, which is for the protein synthesis. But this is a general structural formula for all 20. Then why we will get 20 different types? Due to this variable group. Due to this variable group. Now if I will replace this variable group, check on this diagram, this structural formula only, right? Please have a look over there. Right, when you will see here, the first is a glycine. Here what is there? The R group is replaced by H only. Right, this is the simplest amino acid, which you call it as glycine. What do you call it as? Glycine. Now one type of amino acid. Now R variable group you have to replace by methane. Then which type of amino acid is formed? Alanine. Now you replace it by methyl alcohol, that will be CH2OH. Then which type of amino acid is forming? Serine. Like this we are framing so many different types of the amino acids based on what is this variable group. Rest all will be same in all the 20 amino acids, right? One more important thing is shown by amino acid, that will be the zwitterine formation. When you will see this structure of the amino acid, this you say this is a zwitterine form. Zwitterine means both plus and minus is there. Hydrogen of COH group is shifted on the amino group. and it become NH3 plus. But this is happening at pH value, neutral pH value, right? But if you will go with the like acidic or the basic pH value, like for example, if I will pick up for the basic pH value, basic medium is having OH minus, it will take up this H plus and give out one water molecules. So you will left only by minus charge. This structure is formed in the basic pH value. This is formed in the neutral pH value. But if I am going with the acidic medium, if you are keeping this structure in acidic medium, then what will happen? Acidic is having H plus. So this H plus will bind on here and we will get this structure, means plus charge is only there, there is no minus charge. I again repeat, amino acid can show different type of the ionic states based on the which pH medium. If pH is neutral, then zwitterine both plus and minus. If it is basic, then it will remain as minus charge, no plus charge. If it is going to be the acidic, then it is only having plus charge, not the minus charge, and it is interconvertible. This is the special feature of amino acid, and this is very important question based on amino acids, right? Okay, now coming to the next, that how these amino acids are forming the long chain of the proteins. I said you proteins are the long polymers of that. See, when we are having the carbon here, here is hydrogen, here is R, here is COH group, here is NH2. The peptide bond is formed, this is R1, this is R2, here is NH, H, here is H, here is COH. Look to this point very carefully. What is going to happen? There is a loss of water molecule. There is a loss of water molecule, right? So what is going to be formed? There is a formation of this bond, which you call it as CONH bond, means this is the peptide bond, which you even call it as amide bond. Likewise, they keep on bonding and we are getting these peptide bonds and the chain keep on forming. So if the structure is linear like that, that you call it as primary structure of the protein. If it is going to be helically coiled, that you call it as a secondary structure, will be right-handed helically coiled, having N terminal ending into the C terminal. Even for primary, you are having linear again with the N terminal and the C terminal. Why N terminal? Because it's starting from NH2 group. Why COH? Because it last. It is ending into the COOH group, carboxyl group. That's why C terminal. Linear change, which is telling about the sequence of amino acid, primary structure of the proteins, helically coiled, or it is like a beta plated sheets. This one, or alpha helix. These two are which type of structure? Secondary structure. And tertiary structure is what? Please look to this point carefully. This complete peptide chain, I am not counting here R groups. These complete peptide chain you call it as backbone of protein. If it coil upon each other itself, it going to coil upon itself, crisscross on itself, it form the tertiary structure. Hollow woolen ball like structure, which is biologically active and help in the biologically active processes, means they are working as an enzyme there. So enzymes are working in which state? Tertiary stage, right? Quaternary will have more than one polypeptide having the peptide arrangement in different manners. Like if I am saying for quaternary structure, it can be more than one subunits, polypeptide chains can be there. They can be arranged like a cube, they can arrange like a linear manner. Complexity is more here, types of bond will be more. Here even more is there, but with one, but here will be the many will be there. Many subunits, like for example, hemoglobin with 2 alpha and 2 beta chains. Getting that? So what is the backbone of protein? See, I am not saying backbone of nucleic acid. I am saying backbone of the protein. That is the peptide chain what we are getting, and what is hanging from that? RR group. When the backbone is crisscross on each other, 3D structure is formed, that is tertiary. and quaternary is having more than 2 subunits, and they can arrange in cubicle or in a linear manner. Different architecture is there. That is a quaternary structure. If helically coiled or beta pleated sheets are there, this is a secondary, and linear arrangement you call it as primary structure, right? So in that way we had discussed about the first, the classification what we have kept, amino acid and the proteins in short. Now let's talk about the lipids. For the lipids, to remember, they have carbon and the hydrogen, but the oxygen in the formula will be very little, very less. Now what type of lipids we are having all around? Check it from here. It will be like the example given of fatty acids is one category of the lipid. Next is a glycerol. Then is a triglyceride. These are all simple lipids. These are all simple lipids, right? But this one, phospholipid, lecithin. See, the word is lecithin. It is not lactin. Lactin is a secondary metabolite, fine. But lecithin is a phospholipid. This is what? Conjugated type. Conjugated means with carbon, hydrogen and oxygen. You will find phosphorus here also, other element also. That is conjugated type of lipid. Cholesterol, you call it as a derived type of the lipids. So 3 categories they have given in your book: simple, conjugated and the derived type. What is the simple? Fatty acid. What are the fatty acid? These are the long chain of COOH group, where the R can go with the carbon number 1 to 19. Right? If I am taking the palmitic acid, can you see they have written CH3, CH2, bracket 14, COOH, 14 plus 1, 15 plus 1, 16, 16 including COH. If I am giving you the example of arachidonic acid, it is having carbon 20. But how come? Here they have given R should be up to 90. How 20? Because it is including the carbon of the COOH group. This is very important question. The word including, excluding is very important on this point. So if I am saying palmitic acid, how many total carbon? 16. It will be 16 by including the carbon of COH group. So do remember one more fact. This palmitic acid is an example. If I am saying palmitic acid, this is an example from the saturated fatty acid. What are saturated fatty acid? Which is having carbon-carbon single bond only. This is an example over there for saturated. Whereas example arachidonic acid is an example of unsaturated fatty acid. Unsaturated fatty acid, which is having no doubt carbon-carbon single bond, but they can have double bond. But in arachidonic, total 4 double bonds are there. That you call it as unsaturated fatty acids. So one example in your book they have given for saturated, one is for unsaturated fatty acids, right? So if we are having unsaturation, definitely in chemistry maybe you have learned, it will try to have the lower melting points, and if it is going to be saturated, the melting point is going to be higher. So can I say those who are having higher melting point, they will remain as a solid, whereas which is having low melting point, they will remain as a liquids. So the unsaturated fatty acids will try to remain as a liquid, where the saturated one will be solidified at the room temperatures. Just what I want to convey you, one thing they have given in your book, that is of gingerly oil. Gingerly oil, actually again it is an unsaturated fatty acid which will remain liquid at all the room temperatures. All the room temperatures means either winter is even there, it will still remain as a liquid, right? So unsaturated fatty acids are having the low melting point. So among their examples, different melting points are there, but gingerly oil is having the lowest melting point, right? Then for the glycerol, 3 carbon with the 3 OH group. What you must write for this glycerol, even you call it as trihydroxypropane. What you can call it as trihydroxypropane. Why propane? 3 carbons. Why trihydroxy? 3 OH group is there, right? Now this is a triglyceride. Let me to show you how the triglyceride is forming. Triglyceride is formed when I am taking the glycerol, 1 molecule of glycerol, but to that if I am combining 3 fatty acids like this one. Check it out carefully. One on this side I am making. So for this, let me to take the OH group on this side. And here one more fatty acid. Here is one more fatty acid. Now see, 1 glycerol with 3 fatty acid, there is a loss of water molecule. There is a loss of water molecule. There is a loss of water molecule and there is a creation of bond. Which bond is forming? There is a formation of the ester bonds, right? Here actually it will be becoming, just go back to the points again, right? I am taking this once again, R3, C double bond O, OH, COH, this is COH, right? Here even COOH. So I repeat once again, these are the 3 fatty acids. Now to give out the water, being acidic, it will leave the water. Being acidic, hydrogen is given and here OH. Being acidic, H is given, OH. Now look to this bond. What do you call these bonds as? You call these bonds as ester bonds. And what it has created? It has created the triglyceride. Triglyceride is formed from 1 molecule of glycerol with 3 molecules of fatty acids. This is very important question, please do remember. That's why I struggle to write this all the structure which is given on that page as 1, right? Not here, here, this one. This is for triglyceride, right? This is a structure, what I have drawn, same thing here, right? But what is this? This is a conjugated, simply the last one. Instead of last, the fatty acid. Instead of fatty acid, what we have to put here? Phosphoric acid plus choline group. What I spoke? I spoke the word choline group. This is the choline group. What is this thing? Phosphoric acid. Instead of last fatty acid, in the full step, if we are keeping phosphoric acid and choline group, it will become the phospholipid, which is present in the membrane of the, or you can say it is present in the biomembranes or the cell membranes, right? So here phosphate is even there along with carbon and oxygen. That's why it is that type of a that lipid, what you call it as conjugated, right? Last one is a derived type. In the derived type, can you see the cholesterol is there with how many rings? 1, 2, 3 rings. A, B, C and D. This is first important question. Where the OH group is there? With A ring. Where the long side chain is there? D ring. But D ring is pentagonal. A, B, C rings are hexagonal. So how many hexagonal rings are there? 3. How many pentagonal ring is there? Only 1. Getting that? So these are the question what they can ask from the cholesterol. It is having OH group with the A, double bond in B ring and long side chain will be on D. This is important. If I write its molecular formula C27H45OH. You need not to learn but check on the number of carbons, that is 27, and can you see hydrogen is even in good amount but oxygen is little. So being this is a lipid. Understood? So this is about the lipids. Now let's check on the next one, nucleotides and the nucleic acid. Nucleotides are what? In that case, I will say the nucleotide is a combination of pentose sugar. This is which sugar? pentose sugar, which is having the 1' end, 2' end, 3', 4' and 5' also. Being pentose sugar, it should have 5 carbons. But in the ring there are 4, that's why 5th is in a side branch. Now what is attached to that? There will be presence of OH, H, again OHH, being these are sugars, hydroxyl group is there, here is H, here will be CH2OH. On the 2', if I have written OH, that means this is a ribose sugar. If I am not writing this, right? If I am not writing this, means if I am writing only hydrogen, that means this is deoxyribose sugar. Same structure of the ribose and deoxyribose sugar. Deoxyribose sugar is present in DNA. This is present in case of RNA. One difference, right? Next is what? In nucleotide, what is required? In the nucleotide, there must be the presence of the base, right? Now where the base is attached? Base is attached on 1' position. For example, I am drawing the base, that is the adenine. I am drawing the base adenine, which is a purine and having the 2 rings over there. Otherwise purines are even the guanine. Again it is having 2 rings. If I am going for the bases, these are the bases I am talking about. Bases can be purines or it can be pyrimidines. If it is a pyrimidine, then as per DNA, it should be cytosine and even it can be the thymine, right? But in case of RNA, instead of thymine, what they are having? They are having uracil. So bases are categorized into 2 categories, purines and pyrimidines. Purines have 2 rings, pyrimidines having only 1 ring of carbon and nitrogen. I am drawing right now only for the adenine, right? Adenine is a purine, so it is dicyclic. So I am drawing for that. So it is having this type of structure. Carbon, carbon, nitrogen, carbon, nitrogen, again carbon. It is going to associate it like this. Right, here will be the nitrogen, carbon, nitrogen. Fine. It will be like this. Fine. This is a dicyclic. Can you see 2 rings are there of carbon and nitrogen? How can I say this is going to be the adenine? Here will be NH2 group. And how the numbering we have to do? Like 1, 2, 3, 4, 5, 6, 7, 8 and 9. Fine. Now they have double bonds also. Just we are more after completing their main structure. Just the main idea, it is a dicyclic. This is important. Carbon and nitrogen. Position of nitrogen is right. You have to consider that. 2 rings are there. Now this nitrogen is having hydrogen. And here is 1' position on the sugar. Can I say there is a release of water molecule? And now there is a formation of bond. Which bond is forming? Glycosidic bond. This is glycosidic bond. But it is between carbon and nitrogen, CN glycosidic bond. Now 5' is attached to what thing? Phosphate group. Phosphate is having like this type of structure. Now it is going to attach in 5' position. There will be the release of water molecule. Now which bond is formed? This bond you call it as phosphoester bond. This is phosphoester bond, right? Just take care of this structure right now. This is one nucleotide. How come this is one nucleotide? One phosphate, one sugar, one base. If I want to write it simply, now have a look. This is the pentose sugar. Here is a base. Here is a phosphate. Simplest thing. This is going to be one nucleotide. Right? Now for the nucleotide, they want to make a nucleic acid, the polynucleotide chain. How it is going to be formed? Now see here. Focus on this point. Forget this big structure. Pick up only this simple structure over there. Right? I am drawing again on this side. This is one thing. Base is there. Here is a phosphate. Now adjoining one more nucleotide is there. Base and the phosphate. Phosphate is again having one more OH group. On 3' means here 3' is having OH on this side. Again there is a loss of water molecule. Again there is a formation of bond. What is this bond? This is phosphoester bond. But already formed bond is here, even there, phosphoester bond. Now together you call it as phosphodiester bond. If I again simplify, okay, let's see the next nucleotide. Have a look here. This is a base. This is a phosphate of its own nucleotide at 5' position. At 3' position, which bond is formed? This is a phosphoester bond. But already formed bond is phosphoester bond. Then you together call it as phosphodiester bond. So on it is happening. We are getting the long chain of the polynucleotide. And we are having the thing opposite to that, because in DNA it is going to be opposite to each other, anti-parallel. Then this is going to be 1', this is 4', this is 5'. Going opposite to that, like this is bonded at 3', and here is having their own phosphate. Here is its base, and here it is having the phosphoester bond. Here is a phosphate, here is a base. Now two polynucleotide chain anti-parallel. How anti-parallel? Because this is starting at 5', ending at 3'. This is starting from 3', ending into 5'. Because 5' is ending into phosphate, 3' ending into OH group, right? But how they are connected with each other? With the hydrogen bonds. If it is adenine and thymine, then 2 hydrogen bond. Cytosine, guanine, 3 hydrogen bond, or guanine or cytosine, 3 hydrogen bond, right? So this is the polymeric form of the nucleotides, right? Now coming to the last, what is that? That we discussed about the sugars. Monosaccharide are forming the polysaccharide form. Now how they are forming? Let's check on this point. In that form, suppose I am taking the glucose molecule, which is in the form of, or I am saying the glycogen I am taking as a polysaccharide form, right? Now what is happening in this case? There it is, the presence of OH group, like here. Like this. This is the structure of one glucose molecule. It want to form a chain of glucose molecule. Then again next will come as a sugar. Here is even HOH at this 4th position. Let me say the prime position. This is 1' position, 2', 3', 4', 5', 6'. Why 6' total? Because it is an hexose sugar. Now we want to make a long chain because glycogen is a polymeric form which is made up of glucose as a monomeric form. Now if I am taking here HOH, here if I am taking CH2OH, H, rest are all same. Now check that 1 and 4 of adjoining glucose, there is a loss of water molecule. Can I say there is a creation of this bond now? This you call it as glycosidic bond. But this glycosidic bond is combining the carbon with carbon. Can I say COC? And it is between 1 and 4 carbon. That's why you say it 1,4 glycosidic bond. Likewise, they will keep on extending one after the another, right? One after another, they keep on extending and you will get a long chain. If I will show you through your NCERT book, they had given like this. There is a long chain with which type of bond? That is by 1,4 glycosidic bond. But where the branching come, then it become 1,6 glycosidic bond. At branching it will be 1,6 glycosidic bond, but long chain is showing the 1,4 glycosidic bond. Just I have given you the example of the glycogen. Glycogen is made up from the, you can say from the glucose. This is the homopolysaccharide and it is a reserve food material in case of animals, right? Glycogen is a reserve food material in the animals. And if I take for the starch, even it is a reserve food material in plants made up of glucose. But do remember, glycogen will be branched, whereas the starch can be the branched and even it can be unbranched. Both the forms are there, branched and unbranched. And cellulose will be completely going to be unbranched. Again, monomer will be the glucose. You will find cellulose most percentage in the cotton. Even it is in a pulp by which the paper is going to be formed. Chitin, again homopolymeric form, although it is made from the derived or modified sugar N-acetyl glucosamine. Inulin, again it is a homopolysaccharide made up of fructose. The word is fructose, which is a monomer, and the polymeric form is inulin. But do remember, it is inulin, not insulin. If insulin, then it will be a protein. If inulin, then definitely it will be the polysaccharide, right? So in that way we have discussed in a short the biomolecules, the main aspects. Please go through that once, and in a detailed session I have the nucleic acid, one of the video, you can watch that. And that video, why on nucleic acid specially? Because it is linked to the chapter molecular basis of inheritance. So in this way I am closing today's session. We will meet in next session with a new topic. That's all for today. Thank you, students.